So, very good evening, everyone. So, today's session is all about the biological classification. As we know that till now more than 80% species are undiscovered, but 8.7 million species are discovered. But if someone wants to study them, it is very hard if it is not classified well. So, main purpose of classification is easy identification. So, in this session, the content will be covered is the introductory part, two kingdom classification system, three kingdom classification system, four kingdom classification system, five, six, and the all five kingdoms of five kingdom classification systems. So the biology comes from the Greek word bio means life and logi means study. It is a study of all the living things. When we considered a biological, it is all related with the living things and the processes continuing them in them. The classification word means a systematic arrangement in groups or categories according to established criteria specifically. मतलब जो भी criteria को consider करना है उसके basis पे अलग अलग classification होते हैं. When we consider the biological classification system, it is a systematic arrangement of living organisms in groups or the categories based on similarities and dissimilarities. जब हम living organism के classification की बात करते हैं तो उनमें कितनी similarities और dissimilarities है उनके basis पे उन्हें classify किया जाता है. The main purposes of biological classification or any classification system is breaking a subject into smaller, more manageable, more specific parts so that one can easy identification of organism. कोई भी बड़ी अगर हम हीप ऑफ मार्बल्स की बात करें उसमें अलग अलग तरह के काफ़ी मार्बल्स है, अलग अलग कलर्स के हैं, रफ उसकी सरफेस है, कुछ की शाइनी है, कुछ की स्मूथ है. तो हमें अगर उसको कोई ऐसा मार्बल को निकालने को कहे कि जो स्मूथ सरफेस का है और स्पेसिफिक कलर जैसे कि ब्लैक है, तो हीप जिसमें अलग अलग मार्बल्स को एक ही हीप में रखा है उस पर निकालना आसान नहीं होगा. अगर हम उसे इजीली क्लासिफाई करेंगे तो हम उसे इजीली आइडेंटिफाई कर सकेंगे. तो मेन पर्पस क्या होगा कि जो सब्जेक्ट है बहुत लार्ज है उसको स्मॉलर पार्ट में डिवाइड करना. इसकी वजह से कोई भी ऑर्गेनिज्म को हम इजीली आइडेंटिफाई कर सकते हैं. देन स्टडी ऑफ ऑर्गेनिज्म बिकम्स इजी एंड कन्वीनियंट. अगर किसी को कोई ऑर्गेनिज्म पे स्टडी करनी है तो इजी बन सकती है. Then it will helps to establish evolutionary relationship among different organisms. It will also help in establishing the evolutionary relationships. Then the origin of word kingdom. It was originated when Carl Linnaeus introduced the rank-based system of nomenclature into biology in the year 1735. It is the highest rank which was given by the Carl Linnaeus after the super kingdom domain. First super kingdom is domain, after that kingdom comes. The history where the initiation of classification was carried out by the Aristotle. It was completely based on the morphological characteristics and habitat. Aristotle was an European philosopher. Biological classification was given for the first time by him. It was simply based on the morphological characteristics and it was not scientific. It was not scientific because it is simply considering the habitat and the morphological characteristics. This was the classification system given by the Aristotle. He has classified the plants in the three categories that are the plants which are having soft stems, which are having several woody stems, and one woody stems that are the trees. He has classified animals into two categories on the basis of these two criteria that are the where they live means their habitat and the presence of red blood cells. He has classified the animals into three subcategories that are the animals who live on land, on air, in air and under the water. He has classified the presence of blood cells animals that are the animals which contains the red blood cells and which are not containing the red blood cells. Then after Aristotle, Carl Linnaeus came. He gave the first scientific biological classification, provided binomial nomenclature system and taxonomic classification. He has given the binomial nomenclature in which we have to write the any scientific name using genus and species. It was not complete classification yet, as he was the first who gave scientific classification system. He is known as the father of taxonomy. This is the two kingdom classification system gave by the Carl Linnaeus in 1735. He categorized and classified the living organism on the basis of nutrition and mobility. He has classified organism into two kingdoms that were Plantae and Animalia. But there were some drawbacks of this two kingdom classification. He has included all the prokaryotes and eukaryotes in the same group. As prokaryotic bacteria and cyanobacteria were placed in the plant kingdom, it was not specific. Autotrophs and heterotrophs were kept in same group. When we talk about the autotroph and heterotroph, he has placed the fungi in the plant kingdom. As we know that fungi is heterotroph, whereas all the plant are the autotroph. Unicellular and multicellular were kept in same group. He has categorized all the unicellular organism and multicellular organism in the same group. He has put all the protozoan in the animal kingdom. As we know that protozoa are the unicellular organism, whereas animal is multicellular. Some lower organisms have characters of both plants and animals. So when we consider Euglena, it is a mixotroph. It has not get any proper group in his classification system. So after the two kingdom classification system, next classification system was given by Ernst Haeckel in 1866. That was the three kingdom classification system. He has arranged all the organism based on their cellular organization, whether they are unicellular or multicellular. So he gave three kingdom classification system that were Protista, Plantae and Animalia. Protista is a Greek word which means very first. There were also some drawbacks of this three kingdom classification system. When we consider only Protista, he has included walled and wall-less organism together. He has also included photosynthetic and non-photosynthetic organism together. Also, he has included unicellular, filamentous, and mycelial organism together. These were the drawback of the three kingdom classification system given by Haeckel. After him, the four kingdom classification system was given by Herbert F. Copeland in 1938. He introduced a new kingdom named Monera. Monera is a Greek word which means simplest. So, the kingdom provided by the Herbert were Monera, Protista, Plantae, and Animalia. He has also some drawbacks, like only one, like it was still having fungi into kingdom Plantae. As we know the fungi is heterotroph, we cannot include it in the plant kingdom. Then five kingdom classification system that was given by R.H. Whittaker in 1960. He has introduced a new kingdom that he has completely give all the scientific biological classification using five kingdoms that were Monera, Protista, fungi, Plantae, and Animalia. He has considered these following characters like complexity of cell structure, whether the cell is prokaryote or eukaryote, then the complexity of body structure, means the cell is unicellular or multicellular, then the nutrition किस तरह से लिया जाता है, that is the mode of nutrition, whether the organism is autotroph or heterotroph, then the type of reproduction and the phylogenetic relationship. So he has classified organism into main two categories that were prokaryotes and eukaryotes. He has included all the prokaryotes in the Monera kingdom. Then when we talk about the eukaryotes, he has included all the unicellular organism into Protista kingdom. Then multicellular organism were divided further. If the organism is having cell wall if the organism is having cell wall and can perform photosynthesis, then they were included into Plantae, and if they are containing the cell wall but not be able to perform photosynthesis, they were included into fungi. Then if the organism is not having the cell wall, they are the without cell wall, they all were included into the Animalia kingdom. These are the examples of the kingdoms five kingdom, the Monera includes bacteria and cyanobacteria. Protista includes unicellular algae and the protozoa. Fungi involves fungi. Plantae involve multicellular algae. Bryophytes, pteridophytes, pteridophytes, gymnosperms and angiosperms. And Animalia involves porifera, coelenterata, plat- platyhelminths, esquihelminths, annelida, arthropoda, mollusca, echinodermata, and chordata. These are the keywords. Porifera means spore bearer. It is a phylum of aquatic invertebrate, also called sponges. Then coelenterata, it is a Greek word which means hollow body cavity. They contain a hollow body cavity into their body. Examples are hydra and jellyfish. Then platyhelminths, they contains all the flatworms. Platy means flat body. Then means they have a microscopic size. They lack a well-developed coelom and blood vessels. Example is nematode. Then Annelida, it includes all the segmented worms, means their body is completely segmented. Example is earthworm. Arthropoda, it is the largest phylum. It contains all the organism which is having segmented body, jointed limbs, chitinous cells that undergo molting. जो cells होगा वो molting में जाएगा. And their examples are insects and spider, etc. Then the mollusca, it is the second largest phylum of invertebrate. It is a Latin word which means a soft body organism having a soft body. Then echinodermata, it is a Greek word. Echinos means hedgehog and derma means skin. Hedgehog means spines. They are containing a spines on their body. Then chordata, they are having some stage of development. Notochord means they contain a flexible spinal column at some stage of their development, and nerve cord running along the back, a tail stretching above and behind the anus. After the five kingdom classification, the sixth kingdom classification was given by Carl Woese in 1977. He has classified organism into main six kingdoms and gave three main domains that are the archaea, bacteria, and eukarya. Archaea contains kingdom Archaeobacteria, bacteria contains kingdom Eubacteria, and eukarya contains protista, fungi, plantae, and animalia. So we will discuss about the five kingdom classification in detail. First of all, the Monera. Monera contain Archaebacteria and Eubacteria. Archaebacteria further it has example like methanogens, halophiles, thermoacidophiles. Methanogens are the organism which can produce methane. Halophiles are the salt lovers, and thermoacidophiles mainly found in hot springs. When we contain when we consider the eubacteria, they are the true bacteria. The examples are the cyanobacteria, proteobacteria, spirochetes, and gram-positive bacteria. Proteobacteria are the mainly gram-negative bacteria, and it comes from the Greek word which is proteus, means god of sea. It was not come after the genus Proteus, but it is said that it was came from the Greek word which means god of sea. It is capable of assuming different shape, means the organism which are present in Proteobacteria category, they are having different type of shape, different from each other. So first is eubacteria. Bacteria are the sole member of kingdom Monera. When we consider kingdom Monera, all the bacteria can be included in this kingdom. They are most abundant microorganism. Bacteria occurs almost everywhere. They are categorized by the presence of rigid cell wall, and if motile, then which by means of flagellum. Means in this category, bacteria have been classified on the criteria if the organism possesses cell wall or not, and if they are motile or not, whether they are motile or not. If the organism is motile by flagella, then which type of flagellum is present, that all things have been considered in this category. Then hundred of bacteria are present in handful of soil. As we know that a handful of soil can contain millions of number of cell or hundred of number of different type of the bacteria. Many of them live in or on the organism as a parasite. Some bacteria can be present as a parasite as they live inside or on the body of the another cell. The organism belonging to this kingdom do not contain a true nucleus. Means Monera kingdom contain the bacteria which have been involved uh, included in Monera kingdom. They are not possessing a true nucleus. These are the oldest known microorganism on the earth, originated from progenote. We can say the ancestors, the cell which was the ancestor of this kingdom, that was known as progenote. The cell wall is made up of peptidoglycan except mycoplasma. As we know that mycoplasma are the organism that completely lack a cell wall. As they are not having a cell wall, they peptidoglycan, there is no question of presence of peptidoglycan in the cell wall. Then cyanobacteria. The cyanobacteria have chlorophyll A similar to green plants and are photosynthetic autotroph. Cyanobacteria are having chlorophyll A and they can perform photosynthesis. The cyanobacteria are unicellular, colonial or filamentous, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheet. They often form bloom in polluted water body. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. This is the example of nostoc, which is the cyanobacteria. Then when we consider a bacteria and categorize them on the basis of their cell structure, means cell shape, we can categorize them into coccus or bacilli or the vibrio. If the organism is present in round shape, we can say that organism as cocci. If two cocci shaped organism means organism having two circular shape, it can be said as diplococci. Then if a chain of circular cell is present, we can say it as streptococci. Then if four cells are present together in a box like structure, we can say is tetrad. Eight cells are present, which are the cocci. We can say it sarcina. And if cluster of cocci is present, we can call it staphylococcus. When we consider the bacilli, it is a rod-shaped organism. If two bacilli is present, we can call it diplobacillus. If a chain is observed of bacillus, we can say it's streptobacillus. And if it is having rounded almost and having a rod-shaped or shape, we can say is Coccobacillus. When organism is having a comma shape, we can call it Vibrio. Then spiral shaped organism is known as Spirillum. And if more spiral is observed, we can call it Spirocate. If organism has been classified on the basis of flagellation, means the origin and the arrangement of flagella, we can classify organism into four groups. That is the peritrichous. Peritrichous means flagella dispersed randomly over the surface of the cell. Then monotrichous and polar. Monotrichous means only single flagellum is present. Then lophotrichous and polar means with small bunches of flagella emerging from one end. Means the bunches of flagella is observed at a single end of the bacteria. And amphitrechous means flagella at the both poles of the cell. Two flagella can be seen at the both end of the cell. And the new category is included, that is the atrichous bacteria, means they are not having any kind of flagella. When we categorize the organism on the basis of mode of nutrition, we can classify it and categorize into three main groups, that are the photoautotrophs, mean photosynthetic autotrophs that produce energy from light. They are autotrophs and can perform photosynthesis. Then chemoautotrophs, they produce energy from inorganic substances like sulfur bacteria. Then heterotrophs, they are the common consumer of the words. Example of prokaryotic heterotrophs including parasitic bacteria that feed off host, and saprobes such as bacteria of decay which feed off dead organism. Heterotrophs mainly involves the saprophytic organism which mainly rely on decaying material for the nutritional purpose. Then on the basis of reactivity with oxygen, if organisms requires oxygen for respiration and it is must for their growing purpose, then that type of organism is called obligate aerobes. If the organism is growing in completely absence of the oxygen, that type of organisms are called obligate anaerobes, and facultative anaerobes are the organisms which happy to use CO2 when available but can survive without it. Sorry, facultative anaerobes means they can survive in the presence of oxygen, but they are also able to survive in the absence of oxygen. Then the next category is the on the basis of gram staining process. As we know that gram staining mainly used for the differentiating the gram positive and gram negative bacteria. This is the steps of the gram staining. First of all, we use the crystal violet stain as a primary stain. When we use crystal violet, it stains both gram positive and gram negative bacteria in purple in color. When we add iodine, grams iodine, it will form CVI complex, that is the crystal violet iodine complex, by which this stain converts into brownish in color. After that, decolorization steps achieved by which this CVI complex can be removed from the gram negative bacteria. The main principle is as gram positive bacteria are having a very high content of peptidoglycan in the cell wall, and gram negative is having less peptidoglycan in the cell wall. When we apply the ethanol, it is easily comes the CVI complex comes easily from the gram negative bacteria as compared to the gram positive bacteria. But if the decolorization step can be used for a longer time, both type of cell can be decolorized. After that, simply we used a counter stain, that is the safranin, for the staining the gram negative bacteria. So this was the gram staining process, which will definitely helping in the differentiating the gram positive bacteria as well as the gram negative bacteria. At the end of the staining process, we can differentiate the cells in the microscope, where the gram positive bacteria observed in purple in color, whereas the gram negative bacteria can be observed in red in color. This is the steps of the gram staining bacteria, as I have said earlier. These are the property of bacteria of gram positive and gram negative. The thickness of the cell wall of gram positive is very higher, that is the 20 to 80 nanometer, and in gram negative it is observed only 10 nanometer. Number of layers in wall, when we consider the gram positive bacteria, it involves a single layer. When we consider the gram negative bacteria, it is having a bilayer, as it is containing an outer layer also. The peptidoglycan content is very higher in gram positive bacteria, that is 50%, and less in gram negative bacteria, that is 10 to 20%. Teichoic acid is present in cell wall in gram positive bacteria. Lipid and lipoprotein content is 0 to 3% in gram positive bacteria, and it is very high in gram negative bacteria, that is 58%. The protein content is absent in gram positive bacteria and 9% present in gram negative bacteria. The lipopolysaccharide content is almost zero, and it is present 30% in gram negative bacteria. Gram positive bacteria are very much sensitive to penicillin, and gram negative bacteria are less sensitive to penicillin. As we know that penicillin is a category of a beta lactam antibody, it will directly affect the synthesis of the cell wall. When the synthesis of cell wall is occurring, it forms the hollow like structure, means the bonding of the molecule linkage is not completely achieved in the formation of wall, and all the content can be will be lysed easily. Then digestion by lysozyme is seen in gram positive bacteria in higher quantity, whereas gram negative bacteria can be digested by lysozyme weakly. Then that was all about the eubacteria. This is all things about the archaebacteria. Archaebacteria, also known as the ancient living fossil. Cell wall of archaebacteria are made up of glycoprotein and polysaccharide. Autotrophs are the organisms which use CO2 in atmosphere as a source of carbon for a process called carbon fixation. All the autotrophs are can be considered mostly all the autotrophs type of organisms are included into this category. The cell wall envelope have a high resistance to antibiotic and lytic agents due to difference in cell wall composition. They have a very different lipid bilayer making up a cell membrane. The RNA polymerase of archaea is very similar to that of eukaryotes. The habitat can be of different type. Based on the nature of their habitat, the archaea are grouped into main three categories that are the methanogens. They are present in the gut of several ruminant animals such as cow and buffalo, and they are responsible for the production of methane that from the dung of these animals. Then the halophiles that are present in salty areas, and thermoacidophiles which can be obtained from the hot springs. These archaeobacteria are having similarities with modern eukaryotes, which are the histone proteins are the present in archaeobacteria. They also show DNA replication, transcription, and translation process, and they are containing a monocistronic operons. When we talk about the polycistronic operons, it means there are availability of gene of clusters which encodes different type more than one type of protein at a single time, whereas monocistronic operon is a gene cluster or a gene sequence that encodes only one protein at a time, and all eukaryotic mRNA are the monocistronics. Now the kingdom Protista. The characteristics of Protista are they live in moist environment. Both type of cellular organization can be seen in this kingdom that are the unicellular and multicellular. Unicellular sometimes forms colonies. Multicellular lack tissues. Multicellular organism lacks tissues. Eukaryotic sometimes the organisms are the eukaryotics. Both autotrophic and heterotrophic organisms are included into this kingdom. Both sexual and asexual reproduction can be observed. Three major groups of protista are animal-like protist, plant-like protist, and fungus-like protist. Animal-like protist. Protozoan means first animal. Cells contains a nucleus. Cell lacks a cell wall. They all are the heterotrophs and can move on their own. The locomotion can be seen in in this category. The animal-like protists further classified by their movement like the Ciliophora, Sarcodina, Sarcomastigophora, and Sporozoa. Ciliophora contains a cilia on the outside of the cell and tiny hair-like projection they used for the movement or also used for the gathering the food and as a fillers. We can say the filler is an organ such as antenna used for tasting things by touch or searching for the food. Then this is the example of the Ciliophora, that is the paramecium. It contains pellicle, that is the tough outer wall. It is having a slipper shape. Then it is also containing oral groove like mouth. It is also having a gullet for the holding of food. It also contains a food vacuole where they digest the food. It is also having anal pore for the removal of its. It is also containing two contractile vacuoles, two nuclei, and they produces by either binary fission or conjugation. Then Sarcodina, they have pseudopoods, that is the false food, extension of cell membrane and cytoplasm is continuously open and capturing food. They have many shells and these cells forms limestone marble. Sarcodina because they are having continuous streaming of cytoplasm. This is the example of Sarcodina that is amoeba. Most familial Sarcodini is amoeba. It is having blob shaped, means they lacks definite shape. It is having contractile vacuole which controls the amount of water present inside the cell and also contains the food vacuole where food is digested. The next category is Sarcomastigophora. Many live in many live in animals. Means this type of protease can be observed in living organism animals. It is having symbiosis relationship. At least one benefit they are present in symbiotic relationship as well as the mutualism. In this type of in this category, spore formation is not observed. The locomotion is observed by the flagella or chiropodia, or sometimes with the help of both, and they reproduce rapidly. It is the example of Sarcomastigophora, that is the Euglena. It is a unicellular organism, contains major organelles found in more complex life. They are found to be present as autotroph or heterotroph. Then sporozoa, all the sporozoa are the parasites. They feed on the cell and body fluids. They form the spores, and they can pass from ticks, mosquito, or other animals to human. Example of sporozoa is Plasmodium. Plasmodium is a parasite and responsible for human malaria. Then plant-like protists. These plant-like protists are the unicellular and multicellular. They can form colony. They also can move by their own. They are found to be present as autotroph, as they make their own food by their own. And 70% of Earth oxygen is produced by plant-like protists. They can also produce pigments, that are the chemicals that produce colors. Six groups of plant-like protists, which involves euglenoids, diatoms, dinoflagellates, red algae, green algae, and brown algae. Euglenoids are in green color, unicellular. They found to be present in fresh water. They are phototrophs but can be heterotroph under certain conditions. They contain flagella, eye spore by which they sense the light, chloroplast, and pericle also. Then diatoms are the unicellular. They have 10,000 living species. They are aquatic. They are having a glass-like cell wall, and diatomous earth is coarse powder that comes from dead diatoms, which is mainly used for toothpaste, car polish, and reflecting paint making process. Then dinoflagellates are the unicellular. Their cell walls are like plates of armor. They contain two flagella. They spins when they moves. It is having a pigment production, so it is a colorful, can glow in dark, and cause red tide, means the tide of sea can be discolored into the brownish red in color, that is called the red tide. Then the red algae, they are the multicellular seaweeds. They live in deep ocean waters and used for ice cream and hair conditioner making process, and used as a food in Asia. Then the green algae, they are the mostly unicellular. Sometimes they form colonies. Few are the multicellular, can live in fresh and salt water and land in a damp places. Damp means having a little amount of moisture, and very closely related to the green plants. Then the brown algae, commonly called seaweed, they can contain brown, green, yellow, orange, and black pigment. They are mainly found as attached to the rock. They are having air bladders. Air bladder is mainly required for easy floating to the surface of the water. Giant kelp can be 100-meter long. That is the largest brown algae, and used as a food thickeners. Then the third category of protista are the fungus-like protists. They are the heterotrophs. They are having a cell walls. Most of them contains flagella and are able to move at some point in their lives. The main three types of the fungus-like protists are the slime molds, water, and downy moles. They can reproduce by the spores. This is the water and downy moles. They are mainly found to be present in water or the moist places. They are having tiny thread-like look like fuzz. They attack fruit crops mainly and cause the Irish potato famine disease. Then the slime molds, the fruiting bodies contain spores. The fruiting body of these protists, they contain mainly spores. At first they look like amoeba, then later they look like mold. Live on moist, shady places, feed on bacteria and other organisms. Then the next kingdom is fungi. The characteristics of fungi is they are the eukaryotic cell, unicellular or multicellular. If they are multicellular, ones does not form tissues. Some are composed by special type of cell called hyphae, and group of hyphae form mycelium. Hyphae are the tubular cell with cellular walls. As we see that hyphae contains a segment area we can say is a septum formation, like this. The tube is divided by the wall, that is the wall present in the hypha. When a group of hyphae packaged together, it will form the mycelium. Then mushroom and moles. Mushroom are formed by mycelium and live on decomposing remains on other living thing. Mushroom mainly formed by the mycelium, right? Mycelium mainly formed by the collection or the togetherness of the hyphae, whereas moles are formed by the hyphae only and live on decomposing remains on other living things. Then the yeast are the unicellular, they are not formed by hyphae or mycelium. They all are heterotrophs. They have three way of nutrition. Fungi have three different way of nutrition. They might be a saprophyte, means they take nutrition from the decaying material. Then as parasites their leaves. Also they shows the symbiotic relationship. They can produce by spore. Some perform asexual reproduction and others sexual reproduction. Then the kingdom Plantae. They are mostly autotroph, eukaryotic organism. They contain cell wall containing cellulose. Mostly land-dwelling organisms, means they are mostly found to be present on the land, and they are foundation of almost all the ecosystem. Kingdom can be classified as follow. If the plant is non-vascular, that can be said as mosses, and the examples are the mosses and liverworts. And vascular, then it may produce seeds or may not produce seeds. If plant is producing seeds and having flowers, that are said to be angiosperm, and if that is not producing flower, that are known as gymnosperm. Example of angiosperms are monocote and plant which has been included in angiosperm can be monocote or dicot. If the plant is not be able to produce a flower, that type of plant is known to be gymnosperm. Example are pine trees, evergreens. And if plant is vascular and not producing seed, that type of mainly plant involves ferns, horsetails, and club mosses. Then the kingdom Animalia. Animals are the multicellular heterotrophic organism, obtains their energy by consuming energy releasing from the food substances. Animals typically reproduces sexually. Animals are made up of cells that do not have cell walls. Animals are capable of motion in some stages of their lives, and animals are able to respond quickly to external stimuli as a result of nerve cell, muscle, or contractile tissue, or both. External stimuli can be any factor which can help to stimulation of the plant. Animal, sorry. The animal kingdom can be furtherly classified on the level of organization of cell and tissue. So when we consider the tissue level or the organ level organization, and if the symmetry is bilateral and the body is without cavity, that type of animal is called as acoelomates. Then if it is having a bilateral symmetry, but it is having a false coelom, it is known as the pseudocoelomates. And same having bilateral symmetry but having a true coelom, that are known as coelomates. The example of acoelomates are platyhelminths. Example of pseudocoelomates are aschelhelminths, and the example of the coelomates are Annelida, Arthropodia, Mollusca, Echinodermata, Hemichordata, and Chordata. When we consider the tissue level organization with having a radial symmetry, it can be classified in Cnetophora and Coelenterata. When we consider the cellular level organization, the main one phylum to be introduced, that is the Porifera, means having a pore. These are the summary of the characters which is simply possessed by these five kingdoms, that were the Monera, Protista, fungi, Plantae, and Animalia. And this is an overview of Animalia kingdom, all the phylum that has been included into Animalia kingdom. Their characteristics is completely introduced into this table. So this was all about the biological classification system.